Hello, everyone. Welcome to the first webinar of the PAPERI Webinar Series. My name is Mariam Youssef. I'll be today's moderator. Thank you so much for joining us for our session on exploring target disease associations with the Open Targets platform. I'm delighted to introduce our esteemed speaker, Dr. Annalisa Buniello and Ms. Helena Cornu. Dr. Annalisa is a product manager for Open Targets. In this role, she oversees the development and delivery of Open Targets bioinformatics resources, ensuring they meet the needs of the drug discovery community by collaborating closely with users and stakeholders. She joined EMPL-EPI in 2017 as a part of the GWAS catalog team, where she contributed to the expansion of one of the world's most comprehensive databases of genome-wide association studies. Before this, she conducted predoctoral and postdoctoral research in genetic of sensory function at EMPL, Rome, the Wellcome Sanger Institute, and King's College London. Her research focused on understanding the genetic mechanisms underlying sensory traits through characterization of hearing-impaired mouse models. She holds a PhD in medical genetics and bachelor's degree in pharmaceutical chemistry from University of Naples in Italy. Ms. Cornu is a communications lead for Open Targets. In this role, she's responsible for both internal and external communications, including running the Open Targets social media accounts, writing and commissioning content for the blog, organizing and delivering training and coordinating newsletters and press releases. Helena previously worked in the lab of David Lyons at the University of Edinburgh, researching myelin and multiple sclerosis in zebrafish. She left the bench to join Open Targets and pursue her interest in science communication and public engagement. By the end of this webinar, you'll be able to first define the Open Targets platform data model and identify use cases for the platform. Second, identify the types of target disease, association, and annotation data integrated into the Open Targets platform. Third, describe how the platform uses variant information from the GWAS studies to support target disease associations. Fourth, recognize how the association on the fly and target prioritizations views can be used to facilitate target discovery. So please join me in welcoming Dr. Annalisa and Ms. Helena. Please note that we will have a live Q&A session at the end of the webinar. You can ask the question you need using Ask a Question feature to submit your question. Dr. Annalisa and Ms. Helena, the floor is yours.
Thank you very much for the introduction. That's super lovely. Yeah, I didn't realize we were the first webinar in the series. That's very exciting to be introducing this. So thank you very much for joining. So I'm Helena. I'm joined by Annalisa. And the idea of today is to kind of introduce you to Open Targets and specifically the Open Targets platform. We have divided this up into modules. So this is the outline. And at the end of each module, we'll stop for questions and make sure everyone's on the same page. And so please feel free to ask your questions as we go. We'd much rather you ask your questions so that we can answer them in context. And you can also use the chat function. We'll make sure that we monitor the chat so that we can check if there are any questions. But yes, so I will kick us off by talking about Open Targets. So you may not know, but Open Targets is actually a whole consortium. So we have the Open Targets platform, but behind that is a whole consortium made up of academic partners and industry partners and a whole research program of which the Open Targets platform is one part. So the idea behind Open Targets started because our founders had this conversation about how difficult it is to actually get a drug approved. So from the hundreds of different clinical candidates, actually only a handful get approved every year, and so many of them fail along the way. And we kind of think this is mostly due to safety or efficacy reasons. So basically, the drugs just aren't efficient as efficient as we think they are, or they're not safe enough in humans. And so the founders of Open Targets set about to think, okay, how can we fix this problem? And the way that drug discovery is traditionally done is you start off by trying to identify a target, and then you maybe design a molecule for it, you test it, you see whether it's working, and then you demonstrate clinical value. But at the start of all this is actually picking the target and making sure that you have something that you can drug within that might help with your disease. So that's sort of where we came about. So Open Targets was founded to try and address this problem. And around the time, there was a lot of research showing that if you had genetic evidence that supported a link between your target and your disease, you were much more likely to have a successful drug. So there were a few papers published in 2015 which showed that if you had GWAS evidence for your target, your drug was twice as likely to succeed. And this has been followed up by more research over the years, including some research from Open Targets and from researchers within Open Targets, showing that, yes, most of the drugs that are approved are actually supported by genetic evidence, showing that this is a really important aspect of drug discovery, is identifying this genetic evidence and really putting it to good use to try and find the drug targets that will be the most efficient. So Open Targets is an innovative, large-scale, multi-year, pre-competitive industry academia partnership. And we celebrated our 10 years last year. So we were founded in 2014. So we've been going for over 10 years now. And actually the platform, which is what we're going to talk to you about today, is celebrating its 10 years this year. And we're really proud of all the achievements that we've had over the years. But so Open Targets, so I've mentioned that it's this industry academia collaboration. So we have a number of partners. So on the academic side, we have MBI, which is where Annalisa and I are both based. We partner with the Wellcome Sanger Institute, which has amazing capabilities in genetics research. And then on the industry side, we have these 5 partners, so Genentech, GSK, MSD, Pfizer, and Sanofi, who provide the kind of industry aspect and the industry perspective on the research that we're doing and what's most valuable and what's most important at the moment for drug discovery research. And essentially what we do in one sentence is that we use human genetics and genomics data for systematic drug target identification and prioritization. And we're probably going to say those words a lot today because really everything that we do is aimed at helping people to identify links between drug targets and diseases and prioritizing those for future research. So how do we do this? There's kind of 2 streams to what we do. One is new data that's generated by Open Targets. So we have a whole research program that aims to answer kind of the current gaps in the research or the current gaps in the knowledge. And so we generate new data through our research program. We kind of focus on 3 different axes. So our main areas of interest are oncology, immunology, and information and neurodegeneration with a variety of different research models. And then on the other side of this, we have the analysis of existing data, and that's where the Open Targets platform comes in. So the Open Targets platform brings in a lot of different existing data to kind of give you an idea of what is the current research landscape, what is currently known about different diseases, about different targets, and where might be the most interesting to look at. That's my very quick introduction to Open Targets before we move on to talking about the Open Targets platform. Does anyone have any questions right now about Open Targets? Is there anything that you would like me to clarify, or is there anything that you want to know more about? Okay, I'm going to move on, but if there's anything that comes up to you, if there are any questions that come to you, please do feel free to put them in the chat. So just to summarize what I've talked about so far, in case you joined late. So essentially, our whole method, our whole reason for being is based on this idea that target disease associations that are supported by genetic evidence are more likely to result in successful drugs. And that's kind of our mission here. And so Open Targets is an industry academia partnership, and we create and apply evidence to build therapeutic hypotheses. And the therapeutic hypothesis at its most basic is that if you target this drug in this disease, you have a chance of improving the disease. Okay, I'm going to move on to talking about the Open Targets platform and introducing the platform. So There's a slight lag in the slide, so I'm just, yeah. So essentially the platform, which you can access at this URL, so platform.opentargets.org, it's a data integration and analysis tool for systematic drug target identification and prioritization. And what we do, we integrate over 20 different data sources to build up and score evidence for target disease associations. If you access this URL and you go have a look, you can search for a variant, a study, a target, a disease, or a drug. And you can see all the information that we have for all of these, and then you can also see the links that we have between them. The platform is open source, so if you'd like to take a look at our code, you can reuse it for your own purposes. You can access the data through the web interface that you have here, but we also have a different ways of accessing it, like an API, data downloads, Google BigQuery. We are a public data set for Google and also Microsoft Azure, if that's something that you use. And the platform is regularly updated. So actually, one of the reasons that we wanted to have this training now is because a few weeks ago, we released quite a big update to the platform, which brings in variant information and study information and credible set information into the platform, kind of enriching what we already had with a lot of genetic data. And so we thought this would be a nice opportunity to introduce that to you. So very quickly, just because this is kind of how my brain works and I like to think of things in how they relate to each other, I thought I'd talk to you through our data model so you can see as we go through the presentation how the different bits fit together. So at the very basic, at the heart of all of this, is this relationship between targets and diseases. So everything that we do is geared towards building up that evidence that's going to link targets and diseases together. But we also have a lot of other entities that you can look at in the platform. So for example, we have drugs, so you can look up drugs in the platform, and drugs are really interesting because they provide a source of evidence for target disease associations, because you can link a drug to a target via the mechanism of action, and you can link a drug to a disease via the indication. So all of the drug information that we have in the platform helps to support this link between targets and diseases. You then have this whole new section that we introduced a few weeks ago, which is everything to do with kind of genetics and analyzing publicly available common disease data. So you have variants, studies, and credible sets, and we're going to go into more detail about this in the next section. But all of these also contribute to this evidence between target and diseases, because the credible sets will be one of the links between targets and disease entities. So I've mentioned this, but yeah, all of the ones that are in light blue are things that you can search for, which means that if you go to the platform, you can directly type in your favorite variant, for example, into the search bar, or your favorite gene, for example, and that should come up. You can't currently search for credible sets, but everything else can be a starting point for your exploration of the data in the platform. Does that make sense? Is there anything that you'd like me to go over? This is all very theoretical. We're going to show you what this looks like in a second, but is there anything you want me to go over right now? I can't see any questions in the, oh, what kind of data do you have? GWAS data. That's a great question. We have a lot of different kinds of data. So in terms of genetic data, we have GWAS data. We also have a lot of molecular QTL data. And then in terms of data to support target disease evidence, we have kind of 7 different data types. So everything from GWAS data and, for example, gene burden data to literature data to animal model data, pathway data, lots of different kinds. We try to be quite comprehensive if possible. But yes, in terms of genetic data, GWAS and molecular QTL.
But we will cover all this through the talk.
Yes.
So don't worry. You will have an idea after the talk about which data we have.
Cool. So just to recap very quickly, the platform data model is built on 6 different entities. So targets, diseases, drugs, variants, studies, and credible sets. And essentially the platform kind of does 3 things. So you can have you can explore associations between targets and diseases. You can look at the information that we have about these 6 different entities, and you can also analyze evidence from common disease genetics. So that's one of the things that we do now with the latest update. Annalisa, who will talk to you about study, credible sets, and variant data, which is all the new shiny things, exciting things that we have in the platform as of our March release.
Helen, are you happy to go through the slides? You are, right?
Yeah, absolutely. Okay. Yeah.
So hi, everyone. So as Helena said, we have just released a expanded and bigger platform with loads of data that didn't necessarily, they were not necessarily included in the previous releases. Helena, can you please go on with one slide? So Ellen already mentioned, right? Drugs that have genetic evidence, they are supported by genetic evidence, actually have a greater chance to pass clinical trials. And for us, because the platform, we want the platform to be the best target identification platform, we wanted our targets to get the best evidence. For our users to prioritize their targets, right? So we had another resource before, which was called Open Target Genetics, which used to build hypotheses and connection between targeted diseases based on genetic evidence. But then we thought that having one big platform with both data types would make more sense for the community. So this is why in March, we actually merged the 2 platforms so that we have a big platform with genetics data. Next slide, please. And Ellen already showed this, but just for you to have some context, right? So in the last year, I guess, we have worked very hard to expand the platform. And we've done 2 things, like in a nutshell. Very first thing was to redesign a pipeline, a data pipeline, which we called Open Target Gentropy. And this is a pipeline of integration and downstream analysis of GWAS data and also molecular QTL data. So we have worked very hard to be able to colocalize studies, all GWAS against all GWAS or GWAS against molecular QTLs, also fine map studies to get our credible sets, which are the main unit for us to build evidence between targeted disease. So this was back-end, right? So in the background, we have worked very hard to design these pipelines, which are very, very modern, are very, very competitive, like in the market, in the community. I don't think there is anything better at the moment. And then second thing we've done was to build a UI. So to design our database and our web pages in a way that we could show to users the outputs of Gentropy. And we came up with 3 new pages: credible set page, variant page, and study page. Of course, I'm going to give you a demo, but I think it's very nice. It would be nice for you to go through a few slides just to get some context. So first slide, Helena, please. This is a variant page. So in the variant page, we try to gather information on 6.5 million variants. Why do we only or just show 6.5 million variants in the platform? No, all possible million variants that are around and or cataloged in Nomad, because we are talking about a target disease association platform. So we only wanted to show in the platform those variants that have been associated with a trait through a GWAS study, through a molecular QTL study, that it would make sense for users to build therapeutic hypotheses based on the information, right? So for each variant, we have annotation on the allele frequencies from Nomad, which we'll see later. We have description, so a summary of the variant, so whether a variant is a missense variant, is in a non-coding region, whether there is a gene nearby or just falling and like expressed in the same location as the variant, whether we have some phenotypes associated with a variant, and this can be through GWAS studies, molecular QTL studies, but also through other resources. We have pharmacogenetics resources, we have rare genetics resources, so ClinVar that associates through some user submissions, rare variation to phenotypes. So in a way, you will see our variant page really aims for users to get a very big picture of how the variant can actually support a target disease evidence. Then we have, next slide, please. We have a study page, I guess. Yes. So we have, I told you Gentropy is a study integration and downstream analysis pipeline. So we have a few data sources like the GWAS catalog or EQTL catalog FinGen that make GWAS publicly available. I know you know GWAS catalog because you had a webinar from them, so I'm sure you have some background on that. So what we do in our study page is not only, as you can see on the right side, not only to show results, so to show all variants that have been significantly associated to diseases, to the traits, through the particular studies sorted by credible sets. We'll talk about credible sets later, but we also try, and you can see this on the left-hand side, we also try to report as much metadata as possible from the studies. So we, for example, you can see this study was performed on multiple ancestries. This is something that is very important for us. We are trying to expand diversity of our platform. So we are trying to integrate as many diverse studies as possible. So we show an idea on how many ancestries have been sampled in these studies. We also do a QC or summary statistics from all the data sources. You have as information on the author of the study and main data source. So we try to be as comprehensive as possible. First of all, for users to prioritize targets, but also for users to have an idea on how the studies were performed and the samples and the quality of the study. Then we have a third page, which is a credible set page. Now, credible sets. Actually, before that, there is something very important that I put for you. This is a number of studies, both GWAS and molecular QTL studies that we have in the platform, and you can see that independently from numbers, these are very good numbers. I don't think any other resource has so many studies as we do, fine mapped and colocalized. But this is just for you to see that we are trying to expand diversity of our resource, and of course we can do as well as the GWAS catalog does, because GWAS catalog is the main resource that catalogs GWAS studies, and they're trying to expand diversity. So we try to get as many diverse data from them, and we hope that this also by identifying different data sources that are actually diverse, like Japanese biobanks or Estonian biobanks or some of the African biobanks, and setting up collaboration with them, we hope that we can increase diversity of our platform, at least for genetics data, even more. And then the next page, which is the third page before our demo, is the credible set page. So credible sets are very important for us because, as you will see later, we use credible sets to build target disease evidence. But before going there, I just want to tell you that I'm sure you are familiar, but in very simple words, credible sets are the results of GWAS or molecular QTL's fine mapping. And what happens after fine mapping is that we have a group of variants that they are very close to the signal, right, from the study. And this group of variants contains the causal variance with a certain probability. Now in the platform we use 95% probability for our credible sets to be integrated in and used in the platform. But you can find 99% of probability around, or 98% of probability. So this is the probability that that particular set of variants contains our causal variance. So we have a credible set page in the platform. You cannot search by credible set because credible set is a little bit of a philosophical entity. In the platform, it's like a proxy for a locus, but it's not a locus. So for now, users cannot really search by credible sets or locuses. But this is a landing page that you will see users can actually land on and have info on the credible set. So what can you find on credible sets page? You can find all the list of variants in the credible sets. You can also find the results of our locus to gene pipeline, which is I will tell you about this later a little bit more. I have some slides, but locus to gene pipeline is an in-house developed machine learning pipeline that actually links a gene, which is most probably the causal gene, to a locus in the platform. And so through the credible set page, you can actually see which gene is probably causal for that particular trait in the particular locus. And then what we've done, I told you already, with Gentropy, we've done co-localization all against all. So all our GWAS studies find mapped against GWAS study, and also molecular QTL co-localization, as you can see here. So in a way, you will also find and you can visualize whether there are overlapping traits and overlapping variants that are actually associated with different traits, and whether there is, through molecular QTL co-localization, whether there is a functional link between these studies, so these variants. So credible set page is actually a very important page for us then to build therapeutic hypotheses. And going forward, Helena, please. So how do we build evidence, so target disease evidence from this credible set, this unit, which can be a proxy of a locus? So as I told you, we have developed this very nice machine learning method. The method and has been trained, the feature has been trained by using different features, so distance, molecular QTL co-localization, as I told you, chromatin interaction, and VEP, our ensemble, that's an EBI resource that actually predicts variant effect using like different plugins. So what happens, just very simply, in a very simplistic way, we have a locus, we have a gene, and then we can actually, and the locus comes from a study, so we have a trait associated with the locus. So this is how we build target disease evidence from genetics data. And next slide, please. You will see in the demo that we have a very nice association page where we have a list of targets, in this case, associated with a disease. And then we have all target disease associations from GWAS studies, for example, for GWAS data, scored by association score, which is again another unit that in the GWAS that we use in the GWAS in the platform to actually link target diseases in a way that it's quantitative, right? So to quantify actually the association, we have the association score. And to build this genetic evidence, we use gene traits association with an L2 gene score greater than 0.05. Everything will make more sense with the demo. So if you don't have any question at this point, Helena can actually start the demo. Was everything clear? Do you have any questions for me, at this point, or do you want to see the demo first? Is there anything on the chat?
There are no questions on the chat, but did someone have? Yeah.
Maybe the demo will raise more questions. So everything yeah? Was there a question, Jacob?
Hello, I have a question.
I can't hear
Hello,
you.
can you hear me?
Now I can.
Yeah. Go, you had a question?
We can't hear you, unfortunately.
Do you want to write your question in the chat, maybe? I'm not sure.
Yeah, your audio is not working.
Yes, so I'm asking that the GWAS data, so is it that you have SNPs for a specific disease condition?
Yeah, the variants I'm talking about, they are the SNPs. So the variant page is actually a SNP page in a GWAS kind of nomenclature. We have SNP pages, which is our variant page. I don't know if I hear you and I missed your question. Was that your question?
Yes, so my question is, so you have variants for some specific type of drugs and variants also for some specific disease conditions. Is that the understanding that I'm getting?
Yes, totally. So we actually, to be completely detailed, we have SNPs for traits, right, for diseases. And then we translate, we also see whether for this particular disease, there are drugs that Campbell, our drug resource, reported for that particular trait. So we get genetic data to build evidence, and then we have drugs to annotate whether the disease we are talking about is also already reported to have a drug. But you will see this in the demo.
So we only integrate variants that have links to diseases, because otherwise it was just so many. So the 6.5 million variants that you see in the platforms are the ones that will have evidence linking them to a disease.
Does he answer your question, Jacob? I have another question on the chat as well.
Oh, however,
yes, it answers, but does it mean that the drugs SNPs also the drugs targets also come in the form of SNPs or just statistics?
So I think so is your question whether we know whether the drug, a particular drug, targets the SNP, the mutation that the SNP causes?
No, but the question is that, so you have some statistics for the what do you call it, the disease conditions that we know of.
Yeah.
What about the drugs? How does the data come? Is it also in the form of a GWAS summary statistics, or how is the data? How does the data come? How do we integrate them?
Drug data, as I said, they come from Campbell. So we have a resource, a EBI. Helena, we cover this later, but we have a resource, a EBI, which is called Campbell. And Campbell is the catalog, if we can say that, of drugs. So they actually curate all available drugs for trait. They also curate mechanism of action. They curate any kind of adverse events that can, we also have other data sources for adverse events, but in general, adverse events that you can have for these drugs. And we will show you now how drugs and genetics data are interconnected, but our drugs don't come from GWAS. What we integrate from GWAS is actually whether there is a GWAS study or a molecular QTL study that links a particular SNP to gene with the L2 gene to a disease. Drugs come from another source, and then we cross-check and we see whether for a particular trait, which also has a GWAS study, there are drugs. So it's two different feeds. And
Okay.
you will see them now.
Right. Assuming that I'm looking for a particular trait or disease condition, but then I can't find the summary statistics from your platform. If I get it from the GWAS catalog, is it possible to still come in to use the draft data to work with it?
Very good question. So we actually integrate now all summary statistics from the GWAS catalog. So you should be able to find all summary statistics that are in the GWAS catalog in our platform. And then from the association page that we will now show to you, I will show you where you can see whether there is a summary statistics and there is also a drug for a particular trait. So yeah, that's a thing that you can do. You don't need to go to the GWAS catalog. You only need maybe the study ID from the GWAS catalog. Then you can search for the study in the platform, and you will have the information whether the drug for the trait is in the platform. But
and if you had a study from a different catalog that isn't in the GWAS catalog, for example, you could then go and look up in the platform what information you have for those traits or those variants that you have in your study.
But this will be much clearer when we do our demo. We will show you what we mean. So if you bear with us, I think our answer will be clearer. Okay, let's go, Helena. I'm gonna jump to the...
Yeah, do you want to... There was a question in the chat, which was how are rare variants handled?
Yes.
So do you want to cover that now? Yeah.
Yes, I can answer to that. So at the moment, we don't integrate sequencing studies, no genome sequences, no exon sequences. So rare variants as per sequencing studies, but rare identified by sequencing studies, we still don't integrate them. We are finding a way to integrate them, but we don't really integrate sequencing yet. But what we have supporting rare variation is a couple of data feeds, which again you will see in the demo. One is ClinVar. So I showed you that in the variant page we actually have some information about rare variation from ClinVar. So ClinVar is a resource that accepts submission from clinicians about rare variations, so phenotypes, variant phenotypes linked that have been made in rare diseases in the clinic. And we have this kind of rare variation captured in the platform. What we also have, and you will see this on our Association on the Fly page, is some results. We are not a comprehensive resource for burden tests, but we do have some rare variant aggregation tests in our platform. And these are integrated from different resources. AstraZeneca, FIWOS, Regeneron have made available some rare variant aggregation tests, so we have integrated this, and we capture rare variation with a couple of feeds. But if you are talking about sequencing, we are still not doing that. We are trying to think about what's the best way to harmonize sequencing studies and to integrate sequencing studies. I hope I answer your question. There is another one, but I don't understand.
So Stanley's raised their hand. Would you like to unmute and ask your question?
Yeah.
Hello. Thank you very much for your nice presentation. Am I audible?
Yes, you are. Yeah.
Thank you. I just wanted to know how you identify these variants. I posted it in the chart, but looks as if it was not clear. How do you identify the variants to match them with the specific drug? Thank you.
Okay, Helena, do you want to reply to this? Because I feel like I've replied before. Maybe
That's
you
okay.
have a
Yeah.
yeah.
Well, I think so. The main kind of overlap, especially if you're talking about variants, is pharmacogenetics. So we have, as Annalisa explained, we have this whole section which is about analyzing data from GWAS, and then which means we have all these variants now in the platform which have links to diseases. We also integrate data from pharmacogenetics. I think the main source at the moment is PharmGKB, but yeah, that's kind of the main source of pharmacogenetics. And then this will tell us that this gives us a list of variants that have effects on drugs. And so that's kind of where the main overlap is. So the information that we get about links between variants and drugs is coming from pharmacogenetics. You can also,
to drugs. It's a response to drugs more than actually, like efficacy. But yeah, I don't know if there was the question though. I feel like it's again, maybe you're talking about the drugs that target the variant, right?
Thank you. Do you mean, for example, on the display, you have got acetaminophen and you have got some numbers there. You have written GCST 90, for example.
Sorry. So what you're seeing now is the homepage, which these are just suggested things that you could look up, but the ones that are currently on the page are not related to each other necessarily.
Oh, yes, I know they're not related to each other, but I'm just asking, giving an example, if that's exactly what you meant. You identify the variant like the GCST 90, you relate it with acetaminophen, so you know that variant can be treated. That variant is for maybe inflammation, and anti-inflammatory drug could be acetaminophen. Is that how you relate them, please?
Yeah, Helena, I think the question is in the association on the answer is in the association. Okay. So if you please want to go and show an association on the fly page, it's gonna be amazing, Helena. I think all questions.
Sure. Okay.
Yeah.
I can't think of a good one that has. What's a good example for?
You can even go with the ApoE we were studying. Yeah, I don't know if there are drugs there. So your use case, Stanley, is you have a gene, right? And you want to see which diseases are associated with the with this gene, and if there are drugs. So yeah, Helen, I think I'm gonna leave with you the association on the fly, which is part of your workthrough anyway.
Yeah, I think I don't know whether some of this confusion maybe came from the diagram I shared at the beginning. But the drug information that we have is separate from the genetic information, but both of them contribute to this page, which brings together all of the information that we have that will associate a target to a disease. So in this case, we're looking at the target. So this is what we call the associations on the fly page, or the associations page. And here what we've done is we've looked up a target. And what this is showing you is a summary of all of the diseases here on the left that are associated with this target. And all of this is ranked using a score that we developed in-house. So you can see it's a score between 0 and 1, and you can see that the highest scoring disease is coronary artery disease with a score of 0.82.
Are there drugs in this page that we can?
So you can sort the view. So there aren't any drugs for this particular target.
Maybe CFTR is the one.
Sure.
Just to provide.
So on this page, you'll see, for example, all of the. So here I've just clicked on this particular cell, which is showing me that there is evidence from Kemble for the association between this target CFTR and this disease, cystic fibrosis. So I'm looking at this and I can see that there are a number of different drugs which link my target to my disease. So that's kind of one source. So this is showing you all of the drugs for this particular association. If you go onto what we have, so we have this profile page for CFTR, you can also see all of the known drugs for CFTR. so where they have CFTR as a mechanism of action. And you can look at all of the different drugs that we have that are associated to this target. So then you could also look at all of the variants that we have that are prioritized for this target, and then you could link the two together, if that makes sense. So you can look at all your list of variants that are associated with CFTR, and you can have a list of drugs that are used where the mechanism of action is CFTR. I think the other thing that we mentioned was pharmacogenetics. So I don't know if we have any for CFTR. It looks like we do. So this is that's one thing. We then have pharmacogenetics, which is genetic variants that we know are in CFTR that have been associated with drug response. And so this is what this widget is showing. So this is all of the different variants that we know have an effect on drug response that target CFTR. And you can see that a lot of them here are direct drug targets. So for example, I could then look at this specific variant. So if I click on this variant, if you give it a second to load, this is a variant that we have. This will take you to the ensemble page for this variant. It's being a bit laggy, so I'm just going to stick on this page. There we go. But yeah, so you have all the different variants that have pharmacogenetic links to CFTR. But these are not necessarily linked to the GWAS studies that we have. This is a whole separate section. I'm sorry if we've confused this, but I've kind of shown you a little bit of what I was going to show you in the next section. But yeah.
No, Ellen, I think the fact that we have published on genetic evidence having giving more chance of success. I think then there is an expectation that from a variant we say whether the drug targets a variant or not. I think this is what I'm getting from the audience. They wanna know whether a certain drug is targeting a certain variant, which is not necessarily the use case of the platform. This is a little bit independent of our scientific publication, but I can see that's the confusion that there is. They're asking if we can actually visualize the link. I think in the paper there are gonna be many examples, right, which don't necessarily reflect our genetics data in the platform. So there is another question which is very interesting. Do G variants that have known to have association in certain population would have similar effects in another population? So we cannot predict that. We are basing all our associations and our predictions on the studies, right? So if there is a study, a GWAS study that actually analyzes multiple populations and multiple ancestries, we can see whether there is a difference between populations. But if the study has been only performed, and this happens with majority of studies, if studies have only been performed on just on Europeans or just on Africans, we cannot necessarily predict that with our platform. I'm sure there are statistical geneticists that can do that with other tools, but the platform is not a tool to predict that. We can only report whatever has been reported. We have allele frequencies for our variants, so you can see whether one variant is more frequent in a population than another, but the actual effect, we can only predict whatever is being published and reported by other resources, right? They actually do population studies. Yeah. We don't do that. We only report. But that's I guess that's the aim goal of all resources to be able to predict, right? I know there are variants that are more associated. For example, there is like a variant that is more associated to cardiovascular disease traits in Africa more than in European. So these kind of studies have been performed, but this is doesn't necessarily happen for all variants.
There is, so you can, one of the advantages of doing the kind of all versus all comparison is you can see whether there are other sets that overlap, maybe from different studies in a different population. And also, if you look at our study page, you have a section which is called shared trait studies. So basically telling you whether there are, if there is a similar trait that has been studied in a different GWAS, and what they report. So say, for example, you found an interesting association in a specific population, you might be able to find a different study in a different population for the same traits, and then you could maybe go from there. So we try to kind of show you where the overlaps are as much as possible.
So Helena, if there are no more questions, I suggest you go on with your sessions, and then we have a big demo at the end with all use cases, because otherwise if we start with another demo now, we only have like 40 minutes left.
Sure, okay. So give me 1 second. Okay, so just to kind of situate what I'm going to talk about. So I mentioned in my introduction that kind of everything that we do is geared at building up this evidence between targets and diseases. So what I'm going to show you now, which I've shown you a little bit already, but I'll show you in a bit more detail, is that page that summarizes all of that evidence between targets and diseases. And one of the things that I always stress at this point is that the main work that goes into the platform is in harmonizing all of the different sources of evidence that we bring together. So there's lots of different, so I think I said in my first slide, there's over 20 different sources of information that goes into this association page. And so the work of the platform is to kind of harmonize that and to make it make sense so that you can compare different sources between them, and there is some consistency. One of those things which I've mentioned is the scoring. So we have this score to kind of give you a sense of how much information there is for a given data source. And we also, one of the things that we do is we normalize everything to the same ontology. So for example, one resource might call a disease a certain way and a different one might call it a different way. So there is some layer of kind of what's the word? Kind of before it goes into the platform, kind of making sure that we are everything is talking about the same thing. And so if you see evidence for a specific disease, that is the same across all of the different data sources. With that in mind, so I thought I would show you a page, a disease page, because that's kind of the most common use case. So let's say, for example, you're researching Alzheimer's disease. If I can manage to spell it, Alzheimer's disease. And so please excuse the lag. There is a slight lag between me typing things and it appearing on the screen. So if you were to look up Alzheimer's disease, you have this kind of suggested top hit, but then we also show you other entities that might be the one that you're looking for. In this case, I'm going to click on Alzheimer's disease and show you what that page looks like. So this is the associations page for a disease. So what I'm now looking at is I've looked up this disease, Alzheimer's disease, and here on the left, you can see all of the targets where we have evidence to associate them to Alzheimer's disease. And if you look here at the bottom, there's something like over 12,000 different targets for Alzheimer's. And I mentioned this kind of briefly, but basically what this page is showing you is a summary of the fact that there is of what exists, of the amount of information that exists. We are not providing a confidence score of the association. It's not telling you that, if it's really highly scored, it could be as interesting as an association that's quite lowly scored, but an association that has a lower score just has fewer studies done about it, less research that's been done into that specific association. I mentioned that the platform has over 20 different data sources. We've shown them to you here in a column format. So each of the columns represents a different data source that has that might have evidence. So if it's color, if there's blue, that means you do have evidence. If it's white, that means there is no evidence. And just to kind of situate you in case you're not familiar with some of these, we've grouped them into 7 different data types. So the first one and the biggest one that we have here is genetic association. But we also have somatic mutations, which is the next section here. Drugs, so Kemble, we've had a look at this already. Pathways and systems biology, which we've called affected pathway in this case. Literature, so our main source for this is Europe PMC. RNA expression from Expression Atlas, and animal model information from IMPC. So that's kind of the 7 broad categories, but you can see that genetic data really is kind of the most important one that we have. Yes, so we've looked at this a little bit already, but so if you click on any of these cells, it will bring up the information that we have that's powering this specific score. So in this case, you can see for APP and Alzheimer's disease, it's got quite a high score, which you can tell because the blue is darker. So you have a key at the bottom, so the darker the blue, the higher the score. And this is this one that I've clicked on here is summarizing all of the evidence that we get from GWAS credible sets. So this is all the work that Annalisa has presented. This is how it feeds into the platform. So these are all of the evidences that we have that are coming from our GWAS analysis through our Gentropy pipeline. And each of these rows represents a different credible set that supports this association between this target and this disease. And because we have all these pages in the platform, you can then click on any of these and view the specific credible set that's powering this line. One thing I'll mention as well is that you can browse the platform, no problem. If you want to export any of these findings that you have in the platform, you can do so here. So we have some export options on the right-hand corner of the widget. You can also see what the API query is if that's something that you're interested in finding out more. And then so you can do this for individual widgets. You can also export the view. So the associations on the fly view, you have some options for that here at the top. Yeah. Cool.
And
one thing, yeah.
Sorry, Helena, just to go back to the questions earlier. APP is actually a very good example to show that we have GWAS associations, we have some GWAS data supporting association between APP and Alzheimer's disease, and then Campbell, so if you go through the drug column, also says that there are drugs that target APP, they cure Alzheimer's. So in a way, this is what I was trying to say, that in this very big page, if you put all the dots together, you actually have information about GWAS and drugs together. But just from the GWAS data, unless it's pharmacogenetic, which is drug response or like slightly different, this is the only page where you can actually put things together, whether, oh, there is a drug, maybe the drug targets APP through like some genetic mechanism. We don't know this at this point, but it might be right if you dip into the evidence, into the Campbell data. So that's the only way you can do that. Just starting from genetic data, you don't really know whether there is a drug or not if you are looking for action or non-action, drug action on effect on the disease. So this is what I wanted to reiterate.
Yeah. I was going to add about this page that we've tried to make it as modular as kind of you give users as much control over as they'd like. So we've presented it in this way, and we've applied a specific not that one, sorry. We've applied a specific score, for example, a specific weight to the different data sources, but this is something that you can modify yourself. So just as soon as this loads, you have the ability to change the weight of different data sources. So for example, if you wanted to remove the effect of, I don't know, Orphanet on the page, you could do that. So you could bring down the weight of Orphanet to 0, for example, and that will automatically recalculate all of the scores so that you now have a sorted page that does not include any information from Orphanet. You can also include some of the columns if you want them. You definitely want there to be information from gene burden, for example. That's something that you can do. And then you can also, there's a filtering possibility. So say, for example, you want to only have targets that are in the membrane. That is something that you can do. So you could have, so let's say I want to do targets in the membrane. I'm going to add a subcellular location filter. And I'm going to write membrane. So now it's added that as a filter. So now I've narrowed it down from my 12,000 something. I've narrowed it down to 861 targets that are in the membrane. And I can then look at the evidence for those. One other thing that might be quite interesting, you might have noticed it here. So in this advanced filters option, you have this option to upload a list of targets. So say, for example, you have, you've done a study, you've identified a number of interesting genes, and you want to see what kind of evidence there is for those. That's something that you can do here. So I'm going to show you what this looks like. It might be a little slow, I'm really sorry. So if I just give it a second to load onto the screen. So I'm going to do the example that we have here, but you can upload your own list in whatever format you'd like. And I'm going to run this list. The platform is then going to match it to the closest entity that we have in the platform. So if there's an issue with the name, then it'll suggest to you the name that we have that matches the most closely. And you can choose, okay, I actually don't want this one, but I'm going to include the rest. I'm then going to click on upload hits. And actually what you see here is that none of the ones that I've uploaded are in this list. So none of the genes that I have added here have any evidence that links them to Alzheimer's disease. Okay. Was there anything I wanted to say? Okay, I think that's everything I wanted to say about this specific view. Annalisa, was there anything that you wanted to add? I'm going to just get rid of the filters so that we can see the whole view.
No, I think you cover well. I hope it makes sense now with the two views we've presented. I think target prioritization might be next, right?
Yeah. Cool. Were there any questions that anyone had about what I've shown here?
Yes. Can you kindly show how to download the data from here?
Yes, of course. So there's kind of two options. So one would be if you click on export at the top. Super slow. So if you click on export at the top, you have this option to download the data. And it's going to give you these options. But here, what this is going to show you, what this is going to download for you is the table with the list of targets and the scores for each of the different data types. What you can also do is go into if you want the actual data in the widget, you can go into the widget itself. Click on export. And then you have these options to download it as a JSON or a TSV. And actually, the third thing that you can do, secret third option, is we have this page called Data Downloads. Which is all of the different data sets that we have that power the platform. So all of these are available to download. This is quite a big step up from looking at the widgets. But if you wanted to have all of the data, that's an option that you can have here. So you can download it either from the FTP or from our Google Cloud. Yeah. So those would be your download options. I'm going to go back to my associations page. Were there any other questions?
Yeah. So that data that you would downloading is the association between the target and the condition. So in this, let's say APP and then Alzheimer's disease. That's what you'll be downloading.
So here I would be downloading the scores, but if you want to download the actual evidence, I think, correct me if I'm wrong, Annalisa, you'd have to download each widget that has that evidence.
The tables. So if you are interested in just the drugs, you click on one of the Campbell, APP Campbell, and then you download all drugs that are associated with Alzheimer and APP. If you are interested in the GWAS data, you click on the APP GWAS association dot, and then you can download the particular data set. But then, as Helena said, if you go to the download page, you're actually gonna get a sense of everything. You're gonna get the scores, you're gonna get evidence. So depending on your data sets, you're gonna get the whole series, the entire data set. So it really depends on your use case. So in your case, which data would you be interested, like the scores or the actual evidence?
Well, for now, I mean, any of them. I just want to know how the platform works.
Okay,
yeah.
How to use
it's clear, right? Is it clear how all the data sets are accessible? Yeah, I guess so.
Cool. Thank you for the question. I hope, I think we have a page in the documentation that explains the different ways you can access the data, which
I can actually share it, Helena.
Yeah, sure.
Yeah.
So there's different ways. It kind of depends on how complex your question is and how comfortable you are with programming, because you could also use the API, for example, to pull out all of the evidence for an association, for example. Just checking there are no more questions in the chat. Okay. In that case, I think we'll move on to the other kind of interesting feature of the platform, which is target prioritization. So I'm in this page for Alzheimer's, and you might have noticed here at the top, there's a second option, a second view. So you have target disease association, but you also have this option, which is target prioritization factors. And here what we've tried to do is analyze information that we have about the targets to provide you with an idea of how useful they would be as drug targets. So if I click on target prioritization factors, it's going to show me, so we have a similar structure. So on the left, you have all of the targets that are associated with Alzheimer's disease. But now on the right, what you have is different data that gives you information about how useful a target would be as a drug target. So again, we've divided this into categories to kind of give you a sense of where you are. So the first one would be precedence. So is there existing drugs for this target in the clinic? That gives you an idea of how safe it would be to drug your target. You also have tractability. So what are the different modalities that you could use to target this target? So could you use a small molecule? Could you use an antibody, for example? That's what this section will tell you about. You have this section called doability, which tells you whether there are tools that exist to study this target. So, for example, whether you have similar targets in mice, and so you could use a mouse as a model, and whether you have chemical probes that exist, that means you could try and modulate this target already. And then the final section is safety, which is kind of the biggest one. So safety includes things like, is this a very genetically constrained target? Is it very specific to a tissue, or is it widely distributed? These are all kind of different aspects of target prioritization that we've tried to address in this view. And so here, instead of having a score between 0 and 1, we have kind of traffic light system of whether it's unfavorable, so whether the characteristic that we're looking at is unfavorable for drug targets, or whether it's favorable. So for example, if we go back to our example of APP, we know that there already is targets in the clinic for that target APP, and so it suggests that it's probably quite a safe one to modulate. You can see that it's a secreted protein, so based on the subcellular location. It has orthologs in mice that are very similar, will have a high percentage identity to the ones in humans, for example. So it could, you could use a mouse as a model system. And then one thing that I would like to point out is that this is an analysis that we've kind of thought about and developed based on conversations that we've had with drug discovery scientists. But of course, it's not perfect, and it very much relies on individual people to make assessments. And in general, that's true of the stuff that we present in the platform. So we try to present information to you as it exists and as we find it. But then everything, we always encourage people to kind of look into the original sources and make sure that they agree with what we're presenting. Because here, for example, you can see that there already are targets in the clinic, drugs in the clinic for APP. But you can also see that it scores very unfavorably for mouse models for genetic constraints. It's a highly genetically constrained gene. And you can see that it also scores quite low in terms of tissue specificity and tissue distribution. So these are elements of analysis that we provide to people, but we always encourage people to kind of develop their own thinking about it and decide whether or not they agree with our assessments. And I've shown you the page already, but all of the information that we have here in this target prioritization page is information that you would find in the target profile page. So if we use the example of APP, I'm just going to wait for it to show you. So if we use the example of APP, you can navigate to the profile page for APP. I'm going to click on that. And here, what I'm looking at is similar to the CFTR page that I was showing you before. So this is all the information that the platform has that is specific to APP, but it's disease agnostic. So it's all the information that we have about APP as a gene, as a target, without necessarily relating it to a disease. And all of this is information that you will have seen in the target prioritization view. But what's cool about this is that we have this page for all the other entities. So you could do the same thing for a disease. So for example, I could show you what the page looks like for Alzheimer's disease. So this is our profile page for Alzheimer's disease, showing you the different information that we have. And one thing that I think is worth explaining is this section about ontology. So I mentioned that we use an ontology. So in our case, we use EFO, the experimental factor ontology, as our base ontology in the platform. So all of the diseases that we have will fit into this ontology. And if you don't know what an ontology is, it's how diseases relate to each other. So how different terms are related to each other. So in this case, you can see that all of the diseases that we have in the platform fit into this kind of family tree of diseases that go from very, very high level terms like nervous system disease to very, very specific terms of like really specific disease subtypes. And the reason that I like to spend some time on this is because where in the platform you will see evidence, we also link evidence from a child term of a disease to the parent term. So when I was showing you the associations page for Alzheimer's disease, you could also come across evidence that was for any of the child terms of Alzheimer's disease. So you might see evidence for linking familial Alzheimer's disease to a specific target in the page about Alzheimer's disease. And that's just because we figure that if it's a child term, then chances are whatever evidence there is to associate the child term to the target will also apply to the parent term. If that doesn't make too much sense, just let me know, because I know this is the bit that I always struggle to explain. But basically, the point of this is to try and maximize the use of the data that's in the platform. So we really try to kind of extract as much meaning, as much understanding from all the data that we have in the platform and try to relate it to the other data as much as possible. That's the strength of what we do, because you could go and look up all of this information by yourself, but if you look at it in the platform, we really try to show you how it relates to other other sources of evidence or other data that we have. Cool. If there are no questions, so feel free to ask questions. I'm just going to move over to a drug page just so I can show you what that looks like. So let's say, for example, aducanumab. So again, very similar. So this is a drug page. We have profile pages for targets, diseases, drugs, variants, credible sets, all of the entities that we have in the platform. So again, this is kind of summarizing all of the information that we have about this specific molecule. And I don't have any for this specific drug, but you can see that this is another area where we try to include pharmacogenetics and pharmacovigilance data. So trying to relate, so for pharmacogenetics, trying to relate the drug to any known variants that are known to cause issues with this specific drug. I think that's kind of everything. It's a lot to take in. There's a lot of information in the platform. So hopefully this has given you an overview of the different features and the different ways that you could use the platform and the kind of how to navigate it, how you can go from one page to another. But there is a lot of information in here. So if you have please ask them now. Always available afterwards. If you are exploring the platform and you end up having questions that you're not sure about, please feel free to contact us. I will share a slide at the end with kind of more information and where to find more information about the platform. Oh, Annalisa's put it in the chat already. Look at that. So yeah, so in the chat, Annalisa has put a section of the documentation about data access and the downloads page and the API page for the platform. I'm also going to put a link to the community, which is where you can ask us questions and you can find a lot of other people who've asked questions before and how they've solved their questions. So that's just a good place to explore if you have maybe a use case that's a little bit different and you want to see what other people are doing, or if you have a question about the data that we have in the platform or how to use the platform, that's a good place to go.
And Helen, if it's okay with you,
yeah.
I'm going to share the link with the slides.
Yes, of course.
I already changed the access.
Yeah.
I think it's nice for the audience to go through the slides and metabolize everything we said. So
yes,
I'm going to do this now. And if you have any questions, once you go through the slides again, maybe you can contact us.
So the slides will be in a slightly different order to what we presented today, just because we ended up switching things around, but all the information that we talked about will be in the slides.
There, so you can revisit. And also because we want you to give demos more than present slides, I think having both in your mind is going to be useful.
I think, given there's only about 10 or so minutes left, I think maybe if we just take questions for the last, rather than maybe try and show you some new things, let's just see if there are any questions and see whether we can help you with anything.
Also, if you have specific diseases you would like to see and targets that you like us to explore with you, we have 10 minutes, we can do that. I don't know which field are you interested in, or if you have any specific requests. So that's your time.
Okay, in which case I think we'll probably end there. I'll just show you the last.
Helena, colon cancer.
Oh, there is okay, yeah. Sorry, I missed that. Okay.
Yeah,
so
that's good. Actually, yeah, we might have colon carcinoma. So EFO is a bit. Let's see what we have for colon cancer. Cancer is a very broad. Oh, we do have a colorectal cancer.
So we have, so Annalisa is absolutely right. We have colorectal cancer and we also have colon carcinoma. So this is one of the cases where you might have to dig around to figure out which is the one that, how you would call this in our ontology and how we use that evidence. I'm going to click on colorectal cancer. Just so we see that. But you can see, so because it's a cancer, you now have a lot of information for this section about somatic mutations, which obviously those don't really apply in something like Alzheimer's disease. So you now have a lot more information, for example, from ClinVar or from the cancer gene census.
I think you froze, Helena.
Oh, sorry.
Oh, now you're back. You're back.
I was just, so is there anything specific, any specific data that you wanted to see in colorectal cancer or ask about?
We can start maybe with GWAS studies, right? We have a couple of targets that have genetics evidence.
Yeah, so if I pick the first one, so MLH1, this is based on one study from 2021.
We actually have a favorite target, TP53.
Oh, okay.
Yeah.
So you can look that up directly here. So this is your line for TP53. So we don't have any GWAS associations for TP53, but we do have some from ClinVar, for example. And so again, so one of the things, for example, that we do is we try to add, where possible, kind of extra layers of analyses, for example. So we've included here variant consequence from VEP and from PrepVar, and you have this assessment that someone in our team developed about the direction of effect. So telling you whether the consequence of the target, so whether it's a loss of function or a gain of function, and whether this is a risk or a protective effect for the disease. So here, for example, if I use this line, you can see that it's a loss of function, and this causes a risk for the disease.
Variants there that have been associated by ClinVar with colorectal cancer. I can see they're all on 17, right?
Yeah.
But the changes and variants are all slightly different. So this is telling us that there is a locus there on chromosome 17 that lots of patients have been associated to. They have mutations there, and they have also colorectal cancer. So actually, there is a lot from ClinVar there, and there is like a bit of it was risk, right? A mainly risk.
Yeah.
So there are not GWASs, but there are a few variants that have been associated through clinic with colorectal cancer. And actually, if you dig in to the data, even going to ClinVar links, you might find which patients and how many patients have been studied and sampled for this particular disease. And there are also stars, right? So what ClinVar does, review status, what ClinVar does is also providing some kind of quality control on their data. And for colorectal cancer, it looks like there is 1 star. So they do have a few submissions from clinicians, but it looks like maybe it's, single submitter. It's like maybe only 1 case with this particular mutation. But if you build all cases together, they're all on chromosome 17. So at the end, you can identify a locus, which is not a GWAS locus per se, but it's still something that you can explore. Also lots of literature, right, Helena, on this particular association. So if you go to Europe PMC, you see that there are loads of papers linking P53 to colorectal cancer. So actually, if you go through the literature column, Europe PMC, you can read all papers that link these two entities together.
And you can have a look at, so one of the things that EuroPMC, so the way that we find these papers is a pipeline that we have with EuroPMC to kind of identify matches for the disease term and the target term within a paper. And they have this, there's this way that you can actually see where we found the matches, so you can get a quick idea of whether this paper is actually useful to you by seeing how the two terms are referred to within the paper. Which I think is very cool.
So lots of, you can read a lot. And also final things I wanted to mention, we have a very light blue candle, which means there are drugs, so there might be drugs in clinical trials for...
phase 1.
Yeah, that's it. So there is a drug targeting TP53 for colorectal. Phase 1 terminated, so unfortunately it didn't get through phase 2, only finished at phase 1. It sounds like business or administrative, sounds like they finished money and resources to test this drug. It was a small molecule, maybe someone is gonna repurpose, but there was an attempt to test a drug for this particular indication. And also I was checking, there is, no, that's it. There are animal models. So IMPC is our resource for animal models for diseases. It looks like there are knockouts of TP53 showing colorectal cancer kind of symptoms, or showing the disease, manifesting the disease, which links the target to the disease even more. That's really interesting. You see, they were even testing a drug, and unfortunately, the trial finished. But yeah, this was. Thank you very much. It was a very nice example where you can fix a target, fix a disease. If you have everything clear if you have your question clear in your mind, then you can use the platform just to explore your evidence and to build your hypothesis. And of course, we have on the profile page, right, Helena, we have loads of info on the disease, as Helena was showing earlier.
So I think that's probably a good place to end. And I just wanted to share this final slide with you, just to say thank you for having joined the webinar. And I really hope that you that it was a lot, but I hope that you kind of got the gist of it, understood how the Open Targets platform works. And please do get in touch on the community, or there's our emails at the beginning of the presentation if you have any questions about anything that we've presented today. And yes, I've just included some links here. So if you'd like news from us, you can follow us on social media. We also have a newsletter, and we have these links. So the platform, obviously, the blog where we have release updates and we kind of dive into different areas of open targets. The community where you can ask us questions, and of course the documentation where we've tried to explain everything that we have in the platform, including all the new data. Yeah, thank you very much. I hope this was useful.
Thank you again, Dr. Annalisa and Ms. Helena, for answering the question, for your excellent presentation. It was a pleasure to have you with us. Thank you all for attending. We hope you found it informative and enjoyable. Have a great day, and thank you.
Thank you for having us. Thank you for inviting us. It was a lovely
Thank you. Bye-bye.
Bye. Bye-bye.